Scott, welcome. It's my pleasure to have the opportunity to host Dr. Scott today for Grand Rounds. Hopefully he's coming to us from sunny California, a little bit early there. He originally got his PhD from the University of Saskatchewan in Canada, and then did his postdoctoral work at the Icahn School of Medicine at Mount Sinai in clinical molecular genetics and clinical cytogenetics, and then worked in the Semaphore Clinical and Genomics lab there before moving to Stanford in 2020 to lead the Stanford Medicine Clinical Genomics Laboratory, and is a professor in the Department of Pathology at Stanford University. He came highly recommended to me from my colleague at Pacific Biosciences because I was seeking someone doing awesome work in next generation sequencing, particularly long-read sequencing. And so without further ado, I would like to hand it off to Dr. Scott.
Great. Well, thanks very much for the introduction. Let me just pull up my slides here, make sure. Everybody can see okay?
Yes.
Perfect. Okay, well, thanks so much for the introduction, and thanks for the invitation to present today. It's a pleasure. I visited the University of Minnesota several years ago, and so it's great to be back virtually. I'd like to say hello to Scott Dean as well, too. We went to grad school together, so I hope he's doing well. He was a real, he set the bar high for all of us in grad school, so I hope he's, I'm sure he's doing great work there still. So thanks again for the opportunity to speak. So today I'm going to talk about our work implementing clinical genome-based diagnostic testing. So as far as my disclosures go, so I'm a paid consultant for Semaphore, and I also receive financial support from the University of Puerto Rico for pharmacogenetic research consulting. And for learning objectives, I just put three together to introduce the Stanford Medicine Clinical Genomics Laboratory and to demonstrate the implementation of clinical short-read genome sequencing for germline diagnostic testing. And then, as previously mentioned, also wanted to talk about our work with long-read sequencing. So we'll assess the utility of long-read sequencing for clinical implementation, including pharmacogenomic testing. So to achieve the learning objectives for the outline, I wanted to just really walk through the Clinical Genomics Laboratory and give it a kind of high-level overview of the history and where we're at in the last year. And then talk about our short-read genome sequencing validation work from last year. That was really put together to develop and implement diagnostic panels, and the first ones were for cardiovascular diseases. And then also last year, we also launched a genome-based exome test, and this is all for constitutional disorders, germline. And then finally talk about our work with clinical long-read single molecule real-time sequencing from PacBio as well. So as far as the clinical genomics lab goes, as mentioned, I came from Sinai in 2020, moved the family west during the pandemic. I think I could give a whole grand rounds on that whole experience, but it's been a really fantastic year and a half. The team here is tremendously talented, so it's a real honor to be here. So I thought I would just kind of start with highlighting them. And so in terms of the actual structure of the laboratory, we have a wet lab team. We have a really talented bioinformatics group. They're kind of a hybrid bioinformaticians as well as software developers. And then we have an interpretation team, and that's comprised of PhD level genomic scientists, as well as our genetic counselors. And we also have three additional laboratory board certified co-directors as well that I'll mention later. So in terms of the history of the lab, it actually originated in 2015 through a partnership from SHC and the Children's Hospital. And that was really driven to really drive and stand up a clinical germline NGS laboratory. The goal of having a scalable infrastructure on the IT side, as well as to put together a clinical analysis and interpretation pipeline team. And this was really driven by Ewan Ashley. So I want to acknowledge him and Luann Hudgens from the Children's Hospital side, and many other people who were here before I got here to really help put this together. So this is separate from our molecular pathology laboratory that does all the somatic testing. Our mission is on the germline side. So the original test that was launched was back in 2018, and that was an enrichment-based exome sequencing test, and that was originally outsourced to Personalis. And I'll talk a little bit about that as well a little bit later. And then they also launched a familial variant test, and then also a clinical exome reanalysis test launch prior to my arrival. In terms of the infrastructure, I feel like we are set up quite well now. We have a clinical genotyping platform from Gina. The real workhorses in the laboratory are two NovaSeqs. And then there's a really strong computational team, as well as the infrastructure. This is all done through BaseSpace and Google Cloud Platform, and really sophisticated work from our colleagues in bioinformatics to set this up. And as mentioned, we were really excited in the last year that the hospital supported us on a Sequel II instrument in our clinical environment. So there's also long read sequencing available in independent sequencing cores at Stanford, but this is housed within our clinical environment. So that's what we've been working on this year quite a bit. So in terms of 2021, it was a really fantastic, exciting year. So I'll just kind of walk through some of the development work. I'm not going to talk about all of it. So GIMS is our LIMS system that we're constantly working on through our software team. But the big launch was almost exactly 1 year ago when we completed our clinical genome sequencing validation, as well as our qPCR validation. And that, as I mentioned earlier, that was driven to implement and offer cardiovascular diagnostic panels. And so once we launched that, that basically kicked off a regular weekly run of flow cells generating clinical genomes every week. And then throughout the year, we continued to optimize our workflows, additional validation, software updates. And then last summer, we also validated and launched our genome-based Exome. So this was a very big deal for us too, because as I mentioned, the original Exome here was done by enrichment sequencing. And so we repatriated the sequencing in-house. And then that also resulted in a singular workflow, as well as driving up our clinical genome volume. We also validated the Agena ID panel for this workflow. And then we had a lot of work to do in the fall, winter, because we offer the exome reanalysis test. So we had to reconcile our pipeline to actually take the enrichment-based sequencing data and actually be able to process it through our Dragon-based pipeline. So this was really sophisticated work from our informatics group. So I was really proud of them for that. And then as mentioned too, our long read sequencing platform came last summer. So it's taken us a while to get the resources to get it up and running, but I'll speak a little bit about that later as well too. So that's just been, we see a little uptick in the volume there. And also just mentioned too, really proud of the computational team, because this is a cost savings reinvestment program that we put together for the hospital. So we're basically redesigning our computational architecture to be more cost effective in terms of our storage. And so that we kick that off in the winter now as well. So in terms of our workflows, the idea, the original idea of the laboratory was to have this singular workflow. So we accomplished that last year, which was really, really exciting. And so this is our singular workflow for our short read sequencing panels, as well as our exomes. GIMS is our LIMS system we sequence in-house. This is our Cromwell-based informatics workflow. As I mentioned, base-based, Dragon, Google Cloud platform. We use ELISA for our variant interpretation, and our reporting is done through Mitagen and then sent back to Epic. But in addition to this singular workflow now, we're also actively working on two new workflows. One of them is for our long read sequencing. And so this is in active development right now, using, again, our Cromwell-based informatics workflow, setting it up with SmartLink, as well as some of the new tools for variant identification. And we're evaluating new softwares for interpretation as well. And I'll also mention another workflow that we're chipping away at this year too, which is low-pass genome sequencing. There's a lot of interest in-house for polygenic risk score testing. So we've been chipping away at this. It's an interesting project, validating imputation and things like that, but we'll hopefully have more of this to present in about a year or so, or later this year. So in terms of our dashboard, this is, just our goal is to increase clinical genome sequencing volume at Stanford. And this is basically all of the genomes that have been developed or generated over the last year. And so we can parse them out by specimen type. On the right is blood and saliva is what's really driving our exome and panel-based volumes. And I'm highlighting here the boxes. This is when we launched our exome, where you can see the uptick in the volume of our clinical genomes throughout the year. So I thought I would spend a bit of time, I think for this group, in the pathology and laboratory medicine department, to talk a little bit about the analytical validation work, because the team really did do a tremendous amount of work on the validation. So I thought it would be warranted to present some of that. So in terms of our panel validation, genome panel validation strategy, it's kind of highlighted here schematically. So we did a thorough analytical validation of short read genome sequencing. And as I mentioned, we have a copy number variant confirmation assay using qPCR. And then this is basically the infrastructure to help us and drive our panel validations, which are shown here on the right. And now that we've done this twice, we have a really good architecture and design strategy for it. So it's actually quite efficient now for us to just really build off of that, the genome validation, and then just add on these panels as we identify the clinical indication and the clinical indication and the actual clinical champions to help us identify the need for them. So I don't think I have to explain to this audience about the CAP requirements for validation, but just to kind of highlight all the major elements: reportable range, reference range, specimens, and the validation of test performance, all the analytical work, and the clinical performance characteristics. So I'm not going to go through everything, but I wanted to just kind of, before showing the data, talk a little bit about what data I will be presenting for each, because there is a lot of redundancy. We have hundreds and hundreds of tables of benchmarking data. I don't think I want to spend all the time just showing you those. But what I will present is shown here. So in terms of reportable range and reference range, I will mention that for all three. For specimen and for all the analytical work, I'm just going to present that for the genome validation. And then for the clinical performance characteristics, I will just show those data for the cardio panels as well as for the exome. So reportable range and reference intervals or reference range are kind of vague terms, as probably know, and so they have been redefined for NGS testing. So reportable range, we consider it as the region of the genome in which sequence of an acceptable quality can be derived by the laboratory test. And so for a whole genome assay, the reportable range is essentially the genome. But what we have done in terms of our strategy to look at the genome is to stratify it by different genomic regions. And so we leverage the Genome in a Bottle and GA4GH consortium stratifications that are publicly available. And they basically look at low complexity regions, low mappability, segmental duplications, all difficult regions, not in any difficult regions, and the high confidence regions. So we do all of our benchmarking stratified by these regions to get the most informative look at the test performance. And in terms of reference range and reference interval, we define these based on NGS guidelines as the reportable sequence variations that the assay can detect that are expected to occur in an unaffected population. And so for this, basically our reference interval is small variants and large variants, or copy number variants. So SNVs and indels less than 50 base pairs, and copy number variants greater than 50 base pairs. So I'll start with our specimen validation. And so we accept blood, saliva, assisted saliva, and fibroblasts. So in order to validate these, we had paired samples for all of these different specimen types. And so this is just a look at some of the tables we'll be going through our benchmarking data. And this is non-reference genotype concordance for small variants, SNVs and indels, comparing blood versus saliva, blood versus swab, lymphoblasts, and fibroblasts. And so basically the take-home message is that for small variants, we have very high concordance when we limit it to the non-difficult regions. And also we can look at just the coding sequences throughout the genome to identify this high concordance rate to validate these specimen types. In terms of copy number variants, we stratify by both deletions and duplications, and that we also use both CNV collars and Dragon, the Manta collar, as well as the CNV collar. And then we also stratify these by size ranges. And that's been really important because the performance of both of these CNV collars is quite different based on the size intervals, given the algorithms that are underlie them. And then we also look genome-wide, but also stratifying by the non-difficult regions. And so basically the take-home message is that for deletions, we do a pretty good job of concordance between specimen type. CNV collar has a bit of a known issue with the smaller variants, but luckily Manta does a better job there. So if we combine the two, we feel quite confident in our deletion calling. Duplications is a little bit harder, as you probably know, but we do get good concordance, particularly if you combine both Manta and CNV collar. Manta is a little bit better at the smaller duplications, and CNV collar is quite a bit better at the larger events. So in terms of performance for our genome-based testing, so this is small variant accuracy. So we're looking at all SNVs and indels less than 50 base pairs, and we're benchmarking against the Genome in a Bottle reference materials. And so we have hundreds of tables that look like this, but again, it's just stratifying across all the genomic regions and also stratifying by different variant types, SNVs, insertions of different sizes, deletions, all indels, and all SNVs and indels. So the take-home message, though, is if we look at all SNVs and indels, even in the difficult regions, we're over 99% accurate. But when we look at the high confidence regions, as well as the non-difficult regions, short read sequencing is very accurate at greater than 99.9% accuracy. So as you all probably know, that accuracy is really performance, or accuracy is really driven by the sequencing depth. And so we can look at different depth strata to look at our accuracy. And so that's shown here. We shoot for greater than 40x for our clinical genomes. And this is looking again at the accuracy across different depth intervals. But again, over 40x, we're seeing very high accurate genotype calling. And this can be seen visually in this graph here. So luckily, this is the variant fraction. So the vast majority of the variants that we're covering are greater than 40x. And in the lines are the recall, excuse me, recall and precision that are quite high at that sequencing depth. But as the lower depth strata, you see the reduced performance. So in terms of precision, we're looking at repeatability and reproducibility for our small variants. So I'd like to think we did a fairly robust job at this. We took our reference materials and we subjected them to sequencing in between runs, within the same run, in between instruments, intersides of the instruments. We use S2 and S4 flow cells, as well as the different reagent sets from Illumina 1.0 and 1.5. And again, stratifying genome-wide, as well as just looking at the coding regions. Focusing our view on the non-difficult regions, genome-wide, as well as the coding regions were greater than 99% precise, which was really reaffirming and really a reflection of the wet labs work in terms of their consistency. And this is also shown here if we look at the different types of variants, SNVs and indels. But again, greater than 99% precise across genome-wide, as well as looking at the coding regions. So in terms of CNV accuracy, this is a little harder to do. So there is a benchmarking truth set using NA24385, and we found that to be very useful for deletions. However, for duplications, it's much harder because we found it to be not as robust as a data set as the deletions. So we've only leveraged that truth set for our deletion accuracy analyses. And so as I mentioned before, we stratified by size as shown across the top, and then also by different callers, the MANTA and CNV caller. And so basically our sensitivity, if we combine both, is quite good. We do see some dips in performance at different size ranges, but we felt like this was an acceptable result in terms of our deletion benchmarking. But to supplement this with clinically relevant CNVs, we also took both deletion and duplication samples with known clinical variants, germline variants, as well to do our clinical sensitivity and specificity, which is shown here. So in total, we had 22 clinically significant CNVs. All of them were detected, no false positives or true negatives that resulted in very high sensitivity and specificity when looking at clinically significant variants. So in terms of our precision work, we basically repeated what we did with the small variant analyses, and that's shown here. Again, all of the different comparisons, and we're looking strictly at deletions here, using both MANTA and CNV caller. Not as precise as the small variant calling, but I think that's just the world we live in now in terms of CNV calling from sequencing. But that is also, if we limit this analysis to the non-difficult regions, as just shown at the bottom, our numbers increase substantially, which was reaffirming. Duplications, as I mentioned, is a little bit harder for us. So the numbers are not as strong as the deletions, but when we limit that to the non-difficult regions and combining both callers, we get typically over 90% using one of the two callers in terms of our reproducibility and repeatability. We also did some work for contamination as well as mosaicism detection. And so to accomplish this, we took two genome in a bottle reference materials, NA12878 and 24385, and artificially combined them in the laboratory in different percentages with 12878 as the dominant cell line, and then looked at recall and precision for NA12878. So as increasing levels of another cell line comes in, at what level of that cell line does it negatively impact our variant calling. So in terms of the laboratory contamination that's shown here on the left, Dragon does give a contamination percentage. It's not exactly equivalent to what the laboratory put together, but it is a linear correlation. And so that enabled us to look at our small variant accuracy. And so what we could conclude from this was that just under 10% contamination was acceptable because our accuracy and positive predictive value was still greater than 99%. But anything beyond that resulted in accuracy, a real negative influence on our accuracy calling. So that allowed us to define our QC metrics for contamination levels. We can look at the inverse of this and use that for most, this is the contamination data, just looking at graphically showing here again, after 10%, the recall and precision dramatically drops. So by looking at the inverse of these data and looking at the minor cell line, NA24385, and looking at the positive percent agreement or the sensitivity of that, the minor population, we can use that as an artificial measurement of the ability to detect germline mosaicism. And so to do this, we only look at the sensitivity of informative sites. And so that's a no call or homozygous wild type in the dominant cell line versus a homozygous alternative allele in the minor cell line. And look again at the positive percent agreement. So that's shown here. So again, this is our laboratory mosaicism level, our Dragon contamination level, the counts, and then the positive percent agreement genome-wide, but also limiting that to the coding region. And so graphically, it's shown here. And so basically what we can conclude from this is that at mosaicism percentages of around 30%, we can detect 95% of the variants accurately. And so I think this was very encouraging, but it is a limitation of genome sequencing versus enrichment sequencing, as high depth enrichment sequencing would more likely have an increased capacity to detect low level mosaicism. So from here, I'm going to switch now to the cardiovascular diagnostic panels. So all of that data that we just walked through in terms of that analytical performance, we've also replicated that looking specifically at our cardiovascular ROI, as well as the exome, but I'm not going to show that just to minimize the number of tables that we're all looking at. But in terms of our reportable range or our region of interest or ROI, there are 6 cardiovascular subpanels: comprehensive cardiomyopathy and arrhythmia, comprehensive cardiomyopathy, hypertrophic cardiomyopathy, arrhythmia, aortopathy, and an FH panel. These are the number of primary genes. We also have optional add-on genes. And one of the luxuries that we have using genome sequencing is that we can include non-coding variants as well. And these were identified through ClinVar and HGMD, as well as additional optional add-on non-coding variants as well. And these are limited evidence genes that we can add on for when providers request them. So in terms of our reference interval, so for our diagnostic panels, we actually report out small variants, of course, but also leveraging the genome data and the CNV validation, we also report CNVs as well. So I'm just going to show the clinical performance validation work for this. And so this was accomplished by subjecting known clinical positive control samples that had variants across each of the desired spectrum of variant types. So this is the number of samples per subpanel, the number of genes, the number of variants validated, the different variant types, as well as the different impact they have on the protein, as well as the anticipated classification for all of these different variant types. And so in general, our small variant accuracy, sensitivity, and specificity was all very good. 100% in terms of being able true positives and no false positives. For CNVs, however, we did miss one, and I'll mention that in a minute as well, too, highlighted here. And so because we missed one of the small CNVs, our sensitivity was down to 85%, the specificity was very high, as was accuracy. And so we confirm all of our CNVs by qPCR. And so this is the validation data, just looking at the reported CNV versus the genome-based CNV call, our qPCR results, and the status of that. And so I highlight here the one exon 7 duplication in the LDLR gene that genome sequencing missed. So this was a little bit surprising to us because it has a really strong copy number gain, but it is a small exon level duplication. And so when we look at the IGV, for these are two duplications that we did correctly identify. As you can appreciate, fairly straightforward for the callers to capture these. This is an autosomal duplication 3:2. And this is an X-linked duplication in a male, so 2:1, very straightforward for both MANTA and CNV caller to detect. And this is the LDLR duplication, the smaller variant that we missed. And so if you look at the IGV, if you squint your eyes, you can kind of appreciate the increase in depth at that region. This is zoomed in a bit. And so this is less than 1 kb. And so we're still working on this. And so I think this is a work in progress in terms of optimizing our variant callers for small variant duplication detection. And this is a great example for long read sequencing as well, too. So we have subjected this sample to PacBio sequencing, and it did detect the exon 7 copy number gain for the LDLR gene. So from genome to cardio to exome. So now for our exome reportable range and reference interval. So our Yao Yang from our team designed the exome. And so this is kind of a classic exome, I would imagine. Most of them come from the same sources, all the RefSeq genes, CCDS and GenCode. So it includes all protein coding exons, plus or minus 20 base pairs. And again, though, we also include clinically significant non-coding small variants as well. And we capture those from ClinVar and HGMD. So in total, our exome is about 44 megabases. In terms of the types of variants that we are detecting or reporting clinically, so of course, small variants, we do also look at our larger indels, greater than 50 base pairs. However, we have not included CNV reporting at this stage for our first version of the genome-based exome, as we're still working through the filtration strategy. As you can imagine, there's quite a bit of noise that comes through on that, and so it's a work in progress as well. So again, all of that analytical work from the genome section of the talk, we've replicated all of those accuracy tables, just specifically looking at the exome, but I'm not going to present those. But I will just highlight the clinical performance results. And so those are shown here, similar to the cardio. We were fortunate to capture many different samples with known clinically significant variants, stratified by the different variant types, protein impact, as well as the anticipated classification. And then when we look at our results from that, we also included proband-only analyses, as well as duo and trio analyses. And again, no false positives and no false negatives, resulting in really excellent sensitivity, specificity, and accuracy for genome-based exome testing. So one of the questions, we've presented this to our medical geneticist colleagues, and one of the questions is why? Why bother when there's perfectly fine enrichment exomes available? And so really was the original mission of the laboratory to jump to genome-based diagnostic testing. And so hopefully you can appreciate that it really does simplify our SOP consolidation, having a singular workflow in the laboratory, as well as our bioinformatics workflow, has a singular workflow now, which is really efficient. And then the flexibility in our reportable range. And so in terms of the content, we try to work collaboratively with our providers. And so it allows us to annually update the ROI. We can add genes, we can subtract genes pretty efficiently as new information comes up, as well as optional panel content. Obviously secondary findings, we include the ACMG secondary finding list, but there's a lot of interest internally for incorporating polygenic risk score, as well as pharmacogenomic optional secondary content as well. And then in terms of just the reference range flexibility, as I mentioned, this is kind of in development. We do CNVs for our panels, but however, we have not implemented it yet for our exomes, but that is something we're working on now. And there's new tools always available for repeat expansions and homologous regions. Illumina has some really sophisticated new tools in Dragon that has increased performance on these that we're currently evaluating. In addition to that, there's also just the accuracy. So we found that our genome-based exome at 40x actually has better coverage and improved accuracy over enrichment-based exomes. And so this is the recall and precision benchmarking for our enrichment-based exome. And this is actually very generous numbers. Our previous pipeline was a GATK based pipeline for enrichment exome sequencing. And so this is the data here is our enrichment data processed through our Dragon pipeline. And the Dragon pipeline actually has really stronger performance. But in all categories, the genome-based exome has improved accuracy over our enrichment exome. The only ones that were outliers were due to very small numbers to be able to compare in these samples. So from there, I'll pivot over to our clinical long read smart sequencing effort. And I'll stratify this through kind of three efforts. One of them is Amplicon sequencing, enrichment sequencing, and then finally our stepwise progression into clinical long read sequencing. So our first Amplicon-based work started at Sinai several years ago with my colleague Yao Yang, who joined me here at Stanford. He was the one who really drove the bioinformatics work. And so I just want to start by some of our pharmacogenomic work. And so this is CYP2D6 on chromosome 22, looking at whole genome sequencing data from 1,000 genomes, as well as our own internal genome sequencing data, whole exome sequencing across this region, as well as an enrichment-based panel across this region. And the reason why I show this is because CYP2D6 is historically a very difficult gene to interrogate. And that's because it has these pseudogenes that are highly homologous, 2D7 and 2D8. So of course, genome sequencing is going to sequence across the entire region, but I highlight the enrichment-based tests, the Exome and the PGR-and-seq panel, because those did not intend to capture the pseudogenes. They were designed to capture 2D6. So all of this data here is off-target sequencing. So either the enrichment pulled down erroneously 2D7 and 2D8, or bioinformatically misaligned to that region in a genome. But either way, that results in skewed variant allele fractions within the actual targeted gene of interest. So this was our first target for Amplicon long read sequencing, as shown here on the right. We were able to use PacBio sequencing to really specifically interrogate these difficult regions without any misalignment to any of the pseudogenes surrounding the gene. And so we've published on this a few times, and it's a really straightforward workflow now. Back in those days, we had to do a lot of the informatics work ourselves, but some of the new tools make it much easier. So it's quite straightforward, just barcoding long-range PCR fragments and subjecting them to smart sequencing. We previously did read-based phasing, but now there's a lot more sophisticated phasing tools. But really, one of the real values of a long-read sequencing is to be able to phase these variants across these long reads, which is really important for a lot of Mendelian disorders. So I won't go through all of our work, but we started actually long-read sequencing for methylation detection using smart sequencing coupled with bisulfite sequencing. Excuse me. And then also, as I mentioned, CYP2D6. And then we've also used it for Mendelian diseases and characterizing CNVs. This was a case report of a Bardet-Biedl syndrome patient from Mount Sinai. And then most recently, another really difficult region was the SLC 6A4 homologous promoter region that has a VNTR that short read sequencing just can't even access, but we're able to efficiently cross that with long read smart sequencing. And again, just highlighting the phasing, this is an unpublished work from Sinai. This is a historically difficult Mendelian disease gene region. Just highlighting, there's about 15 or 20 heterozygous variants across this region. That short read sequencing, of course, would not phase. However, 20 kb long-range PCR fragments subjected to PacBio sequencing could really efficiently phase all the variants across that Amplicon. So some more unpublished work on the Amplicon side. So my research interest is in pharmacogenomics, and NUD15 is kind of a hot gene the last few years, implicated in thiopurine response in childhood leukemias. So this was a really great target for us because it's a small gene, about 6 kb. So we were collaborating with PacBio as their chemistry improved. From our earlier work, we were really just multiplexing a small number of samples, but we wanted to use the new chemistry to see how far we could go. And so this was a population survey study of 100 samples from New York, from our Ashkenazi Jewish DNA biobank. And so we could subject all 100 samples into a single smart cell and get excessive sequencing depth. So indicating that we could really, with the new chemistry, include a lot more than 100 samples, probably double that. And this is just the IGV shot to show, again, the phasing. So this is 200 haplotypes, essentially completely phased across the entire NUD15 gene. And so this allowed for, this allowed for complete star allele haplotype architecture, 200 haplotypes in a single smart cell. And we also identified a novel allele during this study that was restricted to the Ashkenazi Jewish population that was not found in any other population. And so this was submitted to the PharmVar consortium, and they adjudicated it and gave it a new number, star 20. And that manuscript is currently in preparation. So in addition to the Amplicon long-read sequencing, we have also been working with PacBio on enrichment-based long-read sequencing. And so this started just as I was moving from New York to Stanford. We started collaborating on this for a program here at Stanford on pharmacogenomics implementation. And so we wanted to use long-read sequencing as an enrichment panel, and not just these amplicons for single targets. And so we worked with PacBio people as well as IDT and now Twist. And so we collaborated on the design. So this is 43 clinically significant genes. 19 of them are full length. It also includes the HLA genes. It's currently about a megabase, but we're actually updating it now. It might probably come in around 2 megabases. And again, subjecting it to sequencing on the Sequel II and leveraging their hi-fi reads that they talk about now, which previously was the CCS reads. And so we presented this at ASHG last fall. And so this is just a couple of examples Again, highlighting the phasing. This is a single sample going across TPMT. You can appreciate the 2 full haplotypes across the entire gene that were captured. For some genes, we just target specific clinically significant variants, as shown here, OPRD1. And again, CYP2B6, which is historically a difficult gene to interrogate. Also, you get beautiful phasing across the whole gene using enrichment-based long reads. So this is fairly exciting to me. I think enrichment using long reads is a really innovative way to do it because you don't have to target the exons anymore, like the short reads. If there's challenging GC-rich regions, you can pull down neighboring regions and the long reads will still capture those difficult regions. So just a couple more examples on the enrichment side. And so as I mentioned, CYP2D6 is a very difficult gene to interrogate. These are 2 benchmarking samples, HG00185 and HG00246. And shown up here are the defining variants for STAR2 as well as STAR4. And so what you can appreciate here is you get beautiful phasing for both samples, which enables us to call the diplotype for this sample is STAR1, STAR2. And for the bottom sample is STAR2, STAR4, based on the phased reads across the entire gene. One of the questions we had, though, was on the structural variation. As I mentioned, CYP2D6 is structurally rearranged quite often as well, too. It has deletions and duplications and gene conversions. And so this is a CNV control for CYP2D6. STAR5 is the deletion. And so again, we were really encouraged to see that using enrichment-based sequencing, you could actually also identify deletions and structural rearrangements of the CYP2D6 region here, showing here the STAR4, STAR5 diplotype really nicely using enrichment-based long read sequencing. Oops. Excuse me, my sound went out for a second. And one of the things, the challenges we have still remaining with the PGX panel is the computational analyses downstream. So I just highlight some of the publicly available tools for doing that translation of VCF into star alleles. And so this is an ongoing area of development as well, too. And so I've been actively working with the Stanford group, Terry Klein, Michelle Corrillo and Russ Altman, using their tool FarmCat that actually takes VCF from any kind of short read or long read sequencing and translates that into the star allele diplotypes. So for the last section, I just want to talk about clinical long read whole genome sequencing. And so this is something we're very excited to be pursuing now as well. And so in terms of why, other than it's expensive, but it actually has, from all the data that's coming out with a lot of the benchmarking work, has shown that Hi-Fi smart sequencing has improved performance, particularly in the difficult genomic regions. And so this is data that's not ours, but it's online. And there's been recent publications as well, too, around this. Looking at benchmarking, this is the FDA Precision Challenge, comparing all the different technologies, Illumina, multiple combined technologies, Nanopore and PacBio, and they stratify it by difficult to map regions, all regions, as well as the HLA, MHC regions, which of course is also historically challenging. And so the takeaway here, if you look at all the different color spectrum, is that combining technologies is actually the most accurate way to sequence. However, if you do not combine, PacBio Hi-Fi reads had improved performance compared to all other technologies. And the numbers are shown here on the right, that using the different participants that submitted these data, in terms of individually comparing platforms, PacBio was the most superior. So in terms of what we're doing with it now, I was hoping to have more benchmarking data available by now to show you, but unfortunately, I don't. We have our plan that's basically all that short read validation that we just walked through. We're going to repeat that whole exercise, but using long read sequencing, and then do a thorough kind of comparison and stratifying it, not by just those genome and abala regions, by a much more broad stratification, looking at GC-rich regions, as well as many other regions of the genome to really identify where the long reads have improved performance. So hopefully we'll have that data later this year. And part of the reason why we're setting up long read sequencing is for some of our translational work. So we're partnering with Dina Madelon from Medical Genetics here. We have a seed grant for the Undiagnosed Disease Network to implement clinical long read sequencing to increase the diagnostic yield in pediatric genetic diseases. And so we're looking forward to kicking that off in the next few months. In addition to the UDN work with Dina, we're also looking to collaborate and support our colleagues from the Gregor Consortium. And so John Bernstein is a really great colleague, and him and Stephen and Matt Wheeler are the PIs of our Gregor site at Stanford. And so John was the PI for the Undiagnosed Disease Network, but as that is kind of winding down, the Gregor Consortium is winding up. And so we're looking forward to supporting them with both clinical short read genome sequencing, as well as our clinical long read sequencing platform over the next 4 or 5 years. So in the last couple of slides, I'll just kind of end with a roadmap for this year and for future landscape. And so there's a lot of more of the same is the goal. We want to continue to drive our short read and long read volume. Lots of development on our LIMS system. The team right now is currently validating a fairly significant update to our germline variant pipeline. Then after that, we've partnered with nephrology. Some really enthusiastic colleagues on the nephrology side who are interested in a comprehensive kidney disease panel. So we have about a 460 gene panel that we'll be validating as soon as the pipeline is ready for launching in the next few months. Also doing SMA screening, but we always want to be developing and validating and launching. So conceptually, I'd like to think we can do that every 3 to 6 months or so. But in terms of what we're actually looking at now, there's ongoing conversations around what panels to bring up. It's a bit of a challenge when we're in the Bay Area, when we have Natera and Invitae and all these other companies around us. So finding niche areas with traction with our colleagues is kind of ongoing strategy work. But Melody Zhang has been scoping out a non-malignant heme panel here for the last year or so. Joan Chow, another laboratory director, is designing an epilepsy genome panel. And again, with our cardiovascular colleagues, there's a lot of enthusiasm for a dyslipidemia genome panel. And I mentioned Yao Yang before. He is the expert on the exome, and so he's actively working on the version 2 already that hopefully will include CNV detection and structural rearrangement detection. We also have a lot of interest on some of the more innovative work, and so we try to balance, developing these things that are actually reimbursable versus those that are not. And so there's a lot of enthusiasm for PRS here, and so chipping away at that this year. I'm personally interested in pharmacogenomics, so hopefully have a panel set up this year. And again, the long reads are so helpful and useful for these challenging regions. There's a lot of neuromuscular colleagues at Stanford that are interested in a repeat expansion panel for the ataxias, Huntington's, Fragile X, and a lot of the other repeat expansion disorders. So I'll end there. To summarize, the Stanford Clinical Genomics Laboratory is committed to developing the infrastructure to support genome-based germline diagnostic testing Short-read genome sequencing can be implemented as a platform for diagnostic panels, clinical exome testing, and it has improved coverage and higher accuracy than enrichment-based exome sequencing. And then finally, long-read smart sequencing is rapidly emerging, in my opinion, as an innovative alternative to short-read sequencing, including germline applications based on Amplicon sequencing, enrichment panels, as well as long-read whole genome sequencing. So I just want to acknowledge the entire organization. The department has been really supportive. Tom Montine is our chair in pathology. I've been talking to him for the last 3 or 4 years or so before I even came to Stanford, and he's been tremendously supportive. And Christina Kong is our vice chair, and Jim Zender is our clinical pathology director. As I mentioned, 3 colleagues, co-directors, Yao, Joan, and Liz. SHC operations, our laboratory team, bioinformatics team, interpretation, and of course also really want to highlight the important support from you and Luann and John Bernstein, who actually now are the advisory committee for the Genomics Lab. So thanks again for the opportunity. It's been a real pleasure. Happy to take any questions if there's time.
Thank you so much, Dr. Scott. That was a great talk. Let's open the floor to questions.
Hi, Stuart. My name is Andy Nelson. I'm one of the pathologists in the clinical genomics lab here as well, where we actually do a combination of both germline and somatic testing. You know, you kind of showed your hand, and I just maybe wanted to have a quick conversation with you again. I mean, the data you showed about the long read sequencing, particularly with, you know, 1 KB or, you know, small resolution copy number gains, it just seems to me that that is going to be the base technology, and that it's going to actually replace short read NGS for germline applications. And just wanted to kind of get your continued thoughts on that and chat with you about that real quick.
Sure. Yeah. I totally agree. So there's some technical challenges we have with long reads, like the DNA input requirements are much higher in terms of like the throughput to do a single smart cell. We have 1 instrument, and so a single smart cell gives you 8x coverage for a whole genome. So that's probably too low. The strategy that PacBio kind of recommends is to do a single smart cell, look at structural variants, and then do another smart cell to double the coverage for the small variants. But that becomes, you know, a little bit labor intensive compared to the NovaSeqs, of course. So, but I totally agree with you. I think, you know, 5 years from now, the long read technology is going to be so superior in terms of those structural rearrangements, the small C and Vs. It's just a matter of getting over that kind of implementation.
Yeah,
and the logistic part. Yeah,
the logistical hump, and basically how we manage the laboratories cost effectively. And
yeah,
that is it.
Yeah.
Well, I certainly don't want to dominate discussion, but I have a different question too. So if no one else is coming online, I'll ask a different question. And it actually goes back to some of the data you showed about the mosaicism and the mixing studies. And, you know, these are things that we do from a similar but very different perspective for our somatic testing, of course, when we're defining what our limit of detection sensitivity is. But it made me think too, we get asked at a not infrequent level, which is a total pathologist term, from our germline colleagues to do quasi-somatic mosaic analysis. In other words, they haven't quite come to terms with some of these congenital anomalies, somatic vascular malformations, and other things are in fact somatic neoplasms or somatic alterations. And they sometimes ask us to utilize our germline platforms, including our whole genome sequencing clinically that we have now for mosaic somatic analysis. And I just kind of wanted to understand too, with everything that you've learned from that careful, you phrased it in some ways as contamination, but really that mosaic analysis with your pipelines for the short read germline NGS, you know, do you have a good feel for where you can capture constitutional mosaicism from those studies?
Yeah, it's kind of challenging because, and we have our wording on our disclaimer, very carefully written, because we don't want to overstate, what we're doing or what we can see, because there's even germline heterozygous variants. The allele fraction can vary widely, so but what we tell people is that we can detect 95% of the variants at 30% mosaicism levels, but we can also detect lower ones, but we just don't know if they're mosaic or not. And so we have to Sanger confirm everything, but that's an ongoing bit of work, though, to try to see if we can drop down the mosaicism detection. And I know in our molecular pathology colleagues are much more aligned with what you're saying too. They have variant allele fractions down to 5%, and that they're trying to detect for the somatic side.
Yeah, no, I mean, we're at a kind of a similar level. I don't think we've been quite as robust in our constitutional mosaicism / contamination detection protocols during our validations as you guys have. But I mean, it's the same thing which we struggle with our clinicians, our geneticists, when they ask us to go after something that we presume ahead of time will be mosaic, and to convey what you just said, that look, if it's less than 30%, we've seen things as low as kind of high teens, 20%. But like you said, I don't really know what the actual accuracy performance metrics are down at that level. I found it very difficult to convey to our
recording stopped.
Yeah,
you don't want to be using this for that purpose.
Yeah, I share your perspective.
Well, thanks, Stuart. I appreciate it.
Thanks, Andy.
I think we're at time, right, Amy? So I guess everyone can get in touch with Dr. Scott if they have more follow-up questions. Thank you again, and we can close out the meeting.
Thanks so much. Appreciate it.